This is TWIS. This Week in Science, episode number 809, recorded on Wednesday, January 27th, 2021. How to describe a mosquito. Hi, everyone. I'm Dr. Kiki, and tonight on the show, we will fill your heads with bad ideas, old milk, and mosquitoes. But first, thanks to the Burroughs Welcome Fund and our Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience. Disclaimer, disclaimer, disclaimer. Just because you want something to be true doesn't make it so. Attacking other people who don't agree with you won't change that fact. For everything you've seen and believe, there are others seeing differently. But some try to see something new by silencing their biases, as much as possible anyway, and taking a scientific view. For everything you see, there is a likely explanation. It may not be the thing you think, but an unexpected reason. So take your preconceptions and throw them in the trash and join us here on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
Good science to you too, Blair, and good science to everyone out there. Welcome to another episode of This Week in Science. We are back again to discuss science because we love it and we know you enjoy it as well. We are minus a Justin this week and hope that his travels are safe and that he will join us again next week after he has landed back in the United States from his adventures in... Where'd he go? Denmark?
Yeah.
went
I
to
don't know why
Denmark.
you would want to come back. Well, I know why he wants to come back here, but also
Why would you want to come back?
the U.S. right now? Oh, well.
Yes. Well, because there's a lot of good going on along with all the rest. And we've got a bunch of science to talk about. I've got one great study out of the United States related to zero carbon future and a plan to get there. This is very exciting. I'm super excited about this story. We also have an interview with one of the world's experts in mosquitoes. And I am thrilled to get to that in a little bit. Blair, what did you bring?
I have a story all about poop. No surprises there. I have another story about poop. So I have two stories about poop in the beginning of the show, just for if you're checking. And then I have two stories in actually the animal corner about CO2 levels and ocean warming. So just all of the climate changey things happening in the ocean and some impacts from that. So,
All right. Sounds like we have a lot of fun science to get to.
Poop and climate change. That's me today.
Woo! Poo positive. As we jump into the show, I would love to remind you that if you are not yet subscribed to This Week in Science, you can find us on Facebook and YouTube by looking for This Week in Science. Also on every podcast directory that's out there, just about. Look for This Week in Science. Look for TWIS Science, TWISCIENCE, on Twitch. And you can go to our website, TWIS.org. TWIS.org to find out more information about the show and get show notes for every episode. Now it's time for the science, so here we go. Carbon zero. Can you just imagine a day? Blair, I want you to put yourself in that headspace of carbon neutrality, where we are taking up as much carbon as we are emitting, where we maybe are not emitting carbon dioxide into the atmosphere. Can you imagine this day?
This is really hard. I feel like I can imagine if we come up with carbon sequestration methods that are really, really big. I could imagine that, but it's really hard for me to imagine a time when we won't be burning fossil fuels at all.
Yeah, it's difficult to even imagine it. But out of Lawrence Berkeley National Lab, there is a peer-reviewed, published roadmap for carbon neutrality for the United States. The researchers have prescribed 8 steps to take in the next 10 years that would get us to that carbon neutrality. So let's dive into what those steps are. Getting to carbon zero, they say, will take the first 10 years. It's pretty much the same, no matter what. Every possible path that they looked at for potential outcomes in the future, the first 10 years, it's all the same. And it's about 2030 when the different paths diverge as to whether or not we're going 100% sustainable renewable energy or whether there is a balanced mix of different types of energy, depending on how much solar, how much geothermal, how much biomass we're using. There are lots of potentials, but in the next 10 years, the 8 things we need to do: increase solar and wind capacity 3.5 times to 500 gigawatts.
That seems doable.
It seems doable.
Mhm.
That's in 10 years. It's not that bad.
Yeah.
Eliminate most electricity generation from coal. Got to get rid of coal. That's a big one. 3, maintain current natural gas generating capacity for reliability. So it's kind of a backup fuel. We can't just get rid of it right now. So over the next 10 years, it's still going to be there, but maybe we start making plans to ease away from it. Number 4, increase zero emission vehicles share to 50%. It doesn't mean that if you have a car right now, you need to just get rid of that car and go buy an electric vehicle. It means that when your car reaches its end of life over the next 10 years, then maybe you consider buying an electric vehicle at that next point. 5, increase sales share of building heat pumps to 50%. Now, what's a building heat pump? It uses the coolness or the constant temperature of below ground to pump heat and cool through buildings so that you don't rely on electricity or fossil fuels for that HVAC system. 6, all new buildings and appliances meet strict energy efficiency goals. So we need to regulate and
that seems like the
get on it.
easiest one, honestly, because that's all new stuff that's being made anyway.
Yep.
Yeah.
If you're building it new, build it clean. Number 7, R&D for carbon capture, sequestration, and carbon neutral fuels. Research and development for the future? Let's do that. Okay, who knew? And number 8, build electricity transmission and pipelines for carbon dioxide and hydrogen gas. Because that is something that we have not done yet, is created infrastructure for hydrogen gas. And if we are going to be moving carbon dioxide from place to place, doing carbon capture or anything else with it, then maybe we need to create infrastructure for these things. Now, the question that you might ask, hey, this is all good, but how much is it gonna cost? Well, over the years until 2050, they say on average it'll cost about $1 per person per day. The total cost will be between 0.2% to 1.2% of our gross domestic product. So it's not going to break the bank. It is a doable amount. And depending on the direction we take, there is actually a net negative path that could be taken that would cost 0.6% of the gross domestic product in 2050.
So I think that's the part of this conversation that always gets me. And without spending the next hour complaining about this, I will just say that whenever we're talking about the cost of shifting to renewable energy, there's a lot of conversation about manufacturing and purchasing and shipping and all this kind of stuff, which makes sense because it's a very tangible cost. But there's the cost of broken down infrastructure, health issues, socioeconomic issues, all sorts of other costs of climate change that you would be reducing by changing it. So it's tough to talk about how much it's actually going to cost until you also figure in what you're saving But I understand that's a lot harder to talk about in the general public and in politics. But it would be great to be able to push that part of the conversation a little bit farther.
It would be wonderful, but the going through this this article I read through it today, and addressing the costs, they went deeply into the fact that they're not asking for energy infrastructure to be destroyed right now and new stuff to be built right now. It's like, okay, we have 10 years to start putting things in the pipeline to take over and to develop the infrastructure to reach the temperature target by 2050. So it's a positive, very realistic view on where we are with energy and where we hope to be. And I was heartened by it, and it was great to see a plan. And this is this is out of Lawrence Berkeley National Lab. So this is researchers tied in also with the DOE and with government programs, so that maybe this is a plan that can really strike some conversations and get some action going. Anyway, I'm excited about it. So I hope other people, take a look at it. We'll put the links on the website. Okay, that was a good idea. Now, Blair, tell me about bad ideas.
Well, I mean, this is my disclaimer. This is my take on this story. So let me just present it to you kind of neutrally, and maybe you can kind of see what I'm seeing here.
Okay, so start from the journalistic, unbiased slant, and then we'll go to the Blair slant. Okay.
So in Australia, there's about 24 million cattle nationwide. Each of them drops about 12 patties a day. That's a lot of poop. There is a huge poop problem in Australia. Bushflies are a huge problem, but on top of that, those cow patties stick around for a long time. Over time, the grass actually rapidly grows around the nutrient-filled cow patties, and that creates what's called rank pasture. It's gross, rank grass that the cows don't want to eat.
It's rank.
So it's rank. It's quite literally rank.
Yeah.
So it would be great to find a way to get rid of this poop. I guess you could just clean it up, but that's a lot of cows. So there's a nationwide dung beetle ecosystem engineers project trying to find a way to get rid of this poop by introducing dung beetles that are active all year round to Australia. There are some species of dung beetles that are native to Australia. They're there during spring and winter. That's when you have really good source for poop, generally speaking, in the natural world, right? But because they need wet poop to lay their eggs in. So that's when they're most active. But they don't really have anything super active from January to April. So they don't have dung beetles taking care of this poop. So they have started introducing other species of dung beetle so that they will then push the cow dung 15 to 30 centimeters down deep into the soil, feed on the dung, and then also it gets turned over back into the soil, and it's a place for them to lay their eggs. So that's the long and the short of it.
Right. Okay. So they need the burrowing dung beetles to take the cow dung, bury it, and use it.
Yeah, and I
Problem
get it.
solved.
Totally, I get it. But I'm just saying, if there's somebody that's going to gung-ho release a non-native animal into their ecosystem, Australia, maybe take a back seat. You've done a lot of this, and it has not turned out well for you at all. So I understand. I don't foresee any problem with this. Dung beetles, as far as I know, they're not going to upset the ecosystem. But we also know that they lay their eggs in poop. So is this going to cause some sort of crazy population boom that is going to impact the ecosystem in some other way? I don't know. But my question is, do they know?
Before any biological control for anything, whether it's a species or piles of dung, you need to take an environmental assessment, right? An ecosystem level assessment.
I'm sure there's been a lot of that done. But there was also a lot of environmental assessment done before other animals have been released into the wild and things haven't worked out that well. And also I'm just saying, Australia, you've had a bad record on this.
So maybe
Yeah.
put a hold on it.
The cane toads and the cats and the rabbits and the non-native fish and the canids and just lots of things. Just
All the
hold off.
prisoners.
I don't know. Maybe clean up the poop. I don't know. I don't know what to do. But I don't have a solution. I'm just raising questions. But moving on from that, I have another really, really cool story about poop.
Okay.
I really wish Justin was here because I feel like he would love this story. So this is looking at white gold, which is seabird guano. Could have been responsible for the highly productive agricultural systems of pre-Incan civilization in the otherwise arid climate of the Atacama Desert.
Wait, what?
Yes.
So gull droppings.
Yes. Yeah. Seabirds of various kinds. But yes, absolutely.
And it's not white gold like my wedding band.
No.
It's
poop.
Guano.
Spirates. Yeah.
Yeah.
So there were these large populations in the Atacama Desert in this pre-Incan civilization in what is now northern Chile between 1000 and 1450 AD. This is one of the driest deserts in the world, but they had robust agricultural systems. And this has defied explanation for a very long time. But what they did is researchers looked at specimens of maize, chili pepper, gourds, beans, quinoa, and wild local fruits from the Atacama Desert between 1000 BC and AD 1800. And they also looked at human isotope values in bone collagen. And they found an increase in nitrogen isotope values from around AD 1000. That shift to extremely high nitrogen isotopes, the highest in the world in archaeological plants. They think this is all based on the use of seabird guano as crop fertilizer. Which has strong implications for their ability to have this very developed agricultural system, the population growth that resulted, and the social complexity that followed.
I mean, agriculture is a big step to that in the first place because you're bulk producing food. But if you have amazing crops because you're using this really nitrogen-rich fertilizer and it's improving the fertileness of the soil, this study is fantastic. I love how they connected the dots also between nitrogen in gull guano with nitrogen in the bones, which would have been isotopes from the food they ate.
Yes, the whole piece, the whole side of science looking at isotopes.
It's like
through
forensic
history.
science. Yeah.
Yes, it is so cool. One of the first times I heard about that was actually bringing it back to climate change from earlier, looking at the different types of carbon isotopes and the ones that come from fossil fuels versus the ones that come from natural processes. And you can actually see based on topsoil, ice cores, all these sorts of things, you can see where the carbon came from because of the isotope. So yes, so just like this, it's like it's like forensics. It's so cool. And I encourage everyone to look into isotopes in soil and fossils and stuff more because it's very interesting.
It's really interesting. Yeah. And speaking of isotopes or leftovers, proteins actually that were left over, I have a study that was published in Nature this week. Ancient proteins provide evidence of dairy consumption in Eastern Africa. So when did we start drinking milk? We must have started to drink milk before we really had prevalence of the lactase gene that allows for the breakdown of lactose, right? Breaks down that milk sugar. And for people who are lactose intolerant, who don't have the lactase gene, we know what that feels like. But African farmers there were herding communities for millennia before there is known prevalence of the lactase gene or of lactase, of this ability to be able to digest it. So they they wanted to find out exactly what was going on. They looked at teeth from individuals in various areas across Africa, dating back as far as 6,000 years ago, and were able to isolate dairy proteins from the teeth and show that, yes, indeed, various groups based on these individuals were consuming dairy, and most likely based on other artifacts and archaeological remnants. They were able to draw parallels between herding communities and individuals that were surviving based on eating dairy before we could digest it. Thanks, teeth.
So, question, how do we know that they were drinking it and not eating it?
They could have been eating it as well. It could have been cheeses. I imagine that it would have been easier or it could have been cultured milks, maybe yogurts, things that soured, fermented a bit in containers. All that they have evidence for are the dairy proteins. So it's not evident as to what form of dairy that was.
Right. Because I'm just thinking certain forms of dairy products are harsher on people who are lactose intolerant than others, right?
Right.
So that if you
depends on the amount of lactose in the dairy that's being consumed.
Right.
Because like sheep and goat milk has less lactose than cow milk.
Uh-huh.
And that's going to make a difference.
Right. Interesting. Right. And then hard cheeses have less than soft cheeses, right? Is that a thing? Okay. So that's what I'm just wondering is if they were consuming it in some ways other than just drinking straight milk right away, and that helped kind of encourage this change. I don't know. Just a curious question.
Yeah, I have no idea, and that would be really interesting to be able to discover. But at the same time, what this is able to say is that before the ability to digest lactose came to be for a lot of people, there were probably mutations around and about for different individuals, but it wasn't widespread. And we know that still within Africa, the ability to digest milk is not as widespread as it is in Northern Europe. And in Germany, it's thought that the ability to digest the genes responsible for creating the enzymes to digest lactose, that they weren't really widely prevalent in the population until about 850 years ago. So that's a long time for these genes to be building up in the population, in the human population. But we've been dairying for a long time. People have put up with a lot to eat dairy for millennia. Well,
it's delicious.
It's delicious. I know. And they probably didn't even have ice cream that long ago. So no. One last quick story before these short stories. 20% of diagnosed autism cases are the result of maternal autoantibody-related autism spectrum disorder. And focusing on this condition, UC Davis researchers at the Mind Institute used machine learning to assess the reactivity of maternal antibodies to 8 proteins expressed in the fetal brain. Now, what happens in the maternal autoantibody-related autism spectrum disorder is that these are autoimmune antibodies where they are thought to be antibodies that are attacking or getting the immune system to attack very specific proteins within the developing fetal brain. And so they looked at 8 of these proteins that are known to be a part of this, and they discovered that there's a very specific pattern of reactivity that they could diagnose with 100% accuracy as to that could be potential biomarkers of autism spectrum disorder. And so they're looking at the possibility of being able to develop a blood test to search for these antibodies, these biomarkers in women before they have children, to potentially give them a better idea of their likelihood to have this specific maternal autoantibody autism spectrum disorder in a child or any other form of autism. But it's potentially good news for women who know that that autism spectrum disorders run in their family. And it can give a little bit more information in this world where we just don't know a lot all the time.
Mm-hmm.
Yeah. If you just tuned in, you are listening to This Week in Science. I'm here with Blair to talk about science with you. And if you're interested in a TWIS shirt or mug or other item of our TWISy merchandise, head over to twis.org and click on our Zazzle store link, and then browse the store to find all sorts of items, some with the TWIS logo and others with Blair's original animal art from the Blair's Animal Corner calendar. And you can buy it and support the show. If you head over to Zazzle right now, peruse while you listen. Okay, at this moment, I'm going to add to our conversation our guest for the evening. Tonight, I would like to welcome Dr. Yvonne Linton. Dr. Linton is a research director for Walter Reed Biosystematics Unit. She holds a trust position within the Department of Entomology at the Smithsonian Institution, National Museum of Natural History. She also serves as curator of the U.S. NM Mosquito Collection, comprising over 1.7 million specimens. It's a lot of bugs. Her first book, Mosquitoes of the World, Volumes 1 and 2, written with Richard C. Wilkerson and the recently deceased Daniel Strickman, was recently published. Dr. Linton, thank you so much for joining us on the show tonight.
Thank you for inviting us. It's great to have this opportunity to talk to everyone here at TWIS, so thank you.
What is it like to be responsible for our nation's mosquitoes?
It's a big job, as you mentioned. It's 1.7 million specimens, so
yeah,
it's definitely something that takes a lot of our time. We're actually a military unit, the Walter Reed Biosystematics Unit, and we've been an integral part of the Smithsonian formally since 1984, but before that, the military, basically when they were starting to work in Southeast Asia and just before the Vietnam War, they were aware that more soldiers were dying of mosquito-borne diseases than any active conflict that was going on. So there started to be a very tight relationship between the military entomologists and the Smithsonian Institution, where all of the expert entomologists were. So that started this relationship between the military and Smithsonian. And at that point in time, there was only 200,000 specimens in the collection. And so just in a very short period of time, we've been able to build it up to the 1.7 million that it is today. And the collection that we have is geographically the most diverse and also taxonomically the most diverse in the world. So we're very proud of it. And it's definitely the jewel in our crown. We go to the collection every day when it's not COVID.
Yeah, when you're able.
Host a lot of visitors when it's not COVID. And even in this COVID time, we're responsible for that collection. So I do personally go and check on the collection, make sure that we're not experiencing any leaks or issues. And we're just working on making it more accessible to people digitally at the moment. So,
of the specimens that you have, I mean, these aren't 1.7 million individual examples of a species. How many duplicates, how many examples of species and subspecies
do we have?
How does it all divvy up within that massive number?
We actually do know how many mosquitoes there have been formally named in the world. That was part of our research for the book, was actually, if you're going to write a book called Mosquitoes of the World, the first thing you need to do is know how many there are, which sounds like an easy ask, but actually it wasn't at all. It took us many, many years to pull together all the information that we needed for the book. But we came to a final figure, and that was that there was 3,570 species that had been formally described and 130 subspecies. So 3,700 dead, which makes it seem like we made it up, but we did actually count every last one. And so in the collection, what we have, the collection was originally started as a US collection. So, it's very heavily US orientated until 1961, when we started to work overseas. And so we did a tally, and in the collection, there's about 65% of all known species. And we have a huge number of the original holotypes, which is the original specimen that the species is named after. So we do have the biggest collection in the world for that. But our collection is comprised of several different forms. We have insects on pins, so we have adults, males and females on pins, but mosquito life cycle is eggs, larvae, and pupae, and the larvae and pupae can't be put on pins. They need to be put on slides. So sometimes we have single specimen where we have a larval skin, a pupal skin, and then we have the adult in the collection. So we have to somehow keep all those things together. So the curation is always moving, always evolving, and we're always realizing that we could have done it somewhat differently to make it easier for ourselves. So
hindsight, it's always better.
Taxonomy, it's a huge business, just doing that work in itself. So we're very lucky, though. Within the unit, we have a lot of people who have worked on mosquitoes for many, many years. We have somebody in our unit just now who's worked for over 45 years in the unit. So we have a lot of history there. We're very, very good at packaging big data sets, maintaining this big data and knowing where everything is. We loan a lot of specimens around the globe and keeping track of that. This person in this university.
Like a library.
It is like a library, but it's really rewarding when you see your collection really starting to shine and really able to find everything that you need. Having visitors there who appreciate the work that you've done on the collection and the expansion that we have. So yeah, it's a very nice dynamic place to work.
And these are all contemporary mosquitoes?
So some of them are mosquitoes that are fossils. Some of them are mosquitoes that have been found once or twice and never found again. So we really don't know where we are in that climb. We just can capture a certain snapshot in time. And an awful lot of our specimens are specimens that are donated to us from researchers worldwide. So if somebody describes a new species, they'll send either the holotype or a few exemplars. Our collections are very strong where we have active military overseas. Everywhere that we are working overseas, we work to protect the soldier. So that's the military aspect is looking at these mosquitoes particularly, but also fleas, ticks, sandflies, any biting insects that can transmit diseases to the soldiers. So really the mission of our unit, the WRBU, is to protect the soldier.
Did you ever envision yourself being a mosquito librarian, a mosquito classification expert, being able to identify and classify via taxonomy all of the mosquitoes?
I think very quickly within my career at university level, I realized that insects were definitely going to be my thing and biting insects at that. So I worked for my PhD, I did culicoides, which spread diseases in agriculture, biting midges. Very small biting midges. And being from Scotland, we were quite famous for our biting midges. So I always knew that I was interested in the disease aspect, in how species could look almost identical. But in fact, when you start to tease them apart, we realized that what we thought was one species can be up to 10 or 12 different species. And when we're talking about controlling particular species which are important in disease transmission, it's really important to know your enemy, to know which of those are actually causing disease outbreaks in certain areas. So a lot of the work that we do is on identification of species. But now it's not only enough to have a microscope and be able to physically look at the specimens. We often need DNA analysis as well. So we've worked very hard at WRBU, working together with the classical taxonomists to make reference libraries of DNA barcodes, which is basically, the way that we're able to rapidly identify and confirm and compare with other scientists that we have exactly the same taxa that we're talking about.
And you've been doing that for a really long time. You spearheaded the mosquito barcoding initiative.
That's right. It's a never-ending story. We seem to discover more new species than we actually get through. So when we first started, we said that we would aim to look at 80% of the world's mosquitoes and get barcodes for those. But the numbers of new taxa that we keep finding everywhere we go is phenomenal, actually. It's about a third extra taxa everywhere that we look. So
wow.
Every group within every country.
So we've been underestimating the diversity of mosquitoes.
Grossly underestimated. And I think one of the aspects is as well that over time, a lot of the mosquitoes have been collected for medical, biomedical studies. So they've really been pulling out those taxa that we already know is important in transmission, rather than looking at everything in the collection. So we've actually underestimated a lot of vectors, important mosquito that can transmit diseases. So a lot of our work now in WRBU is biosurveillance. So we're working, collecting and screening for pathogens. Those pathogens we know and those pathogens we don't. So we're doing a lot of next-gen sequencing approaches everywhere across the globe, really.
Yeah, I just came across a study today talking about a new malaria mosquito that had been endemic to Asia and India. And part of the issue of malaria in the Indian regions, but has recently spread to Africa. And where Africa has had a malaria problem in rural regions, this is now causing a problem in cities because it is a better host for the malaria parasite than it's like, yay, infect me. I love it. It really does well in buckets of water that could accumulate in cities.
And also in water towers as well. People have water just in the top of their houses. So that's where we've been finding it. We actually were involved in the first study that reported that. And actually recently we just had another invasive species that we picked up, this time in the Caribbean. So it's a really good vector of chikungunya, dengue, yellow fever viruses. It's called Aedes vittatus. Very obvious because it has 6 spots on its back like a dice, big white spots. And we picked that up through biosurveillance, actually, activities with the Navy in Guantanamo Bay in Cuba. And at the same time, other colleagues in Dominican Republic have found it in Dominican as well. So that shows, we were able to show, looking at the DNA barcodes, putting biogeographical analysis in there, we were able to show that the 2 populations in the Caribbean were different introductions, but that they both stemmed from the southern Indian continent.
The idea of the introduction, right, is are they brought over in water in shipping containers, or are they carried on the wind? Is there a known method for in banana plants? I don't know. Is there a known method for introduction?
So, there are many, many examples now because the world is so small. We're really globalized, and the number of aircraft that move around, the number of ships that move around, our world is very small now. And these mosquitoes usually don't travel as mosquitoes. They usually travel as eggs. So what we have is a number of different species, usually Aedes species, which are very good at surviving in the dormancy stage as eggs. And so perhaps the mosquito in Southeast Asia lays its egg in a tire or on a lucky bamboo plant. It's shipped as an egg, and as soon as it arrives into USA or any other place in the world, usually if it's tires, they're just dumped out on the dock. And as soon as it rains, the larvae are stimulated to hatch. So it's actually a big problem that we have. We are finding more and more invasive species, but we're looking now. We're in COVID. We're looking at pandemics starting. The issue with these Aedes is often that if the mother is already infected, her eggs will be infected, and so when she moves to a new region, that means that all of a sudden you can have an outbreak of a disease. So, but we also did a very, very nice study with Tovi Lehman from the NIH. And we looked at why malaria disappeared from the Sahel region in Mali. And so basically in Mali, at certain points of the year, it's nice and green and there's lots of water on the ground. And then there's no water, there's no mosquitoes, and there's no malaria. And the question for a long time has been, where do the mosquitoes go? Do they estivate? Where do they estivate? And nobody could find them. We worked together with Tovi doing the molecular identification of mosquitoes that he found on balloons that were put up over the Sahel. So this is actually a picture that was done by our friend and illustrator of the book, Tina Litwack. And actually each one of those mosquitoes is a perfect Anopheles gambier.
That's amazing.
Yeah. So that's just showing, our balloon over Mali with the sticky nets there. And actually one of the things that was shown was that the higher that we were able to put the helium balloon, the more Anopheles mosquitoes that we found. But one of the really interesting things for me was that as soon as I looked at the samples that were brought back to the lab, I noticed that all of the mosquitoes were gravid. That means that they had, within 36 hours prior to being found up in this, high altitude, they had taken a blood meal. This means that it's a strategy. It's an exit strategy. I take a blood meal. Mosquitoes, normally, if they take a blood meal, they just hide out somewhere cool and dark until their eggs are matured. But this is the flight of their life. They take the blood meal, they get up into the airstream and do long distance migrations. And so Tovi's paper that you can see here showed that these mosquitoes could be carried up to 350 kilometers.
And it's just the wind. They're just allowing the wind to loft them and take them up to these heights?
They are. There's some indication that they are actively orientating within the wind with other people's studies. We weren't looking at that in this particular study, but one of the things that obviously is happening in Africa, across Africa, is that people are trying to eliminate malaria. Bill and Melinda Gates, that our senior author of the book, Dan Strickman, worked for, are looking to eliminate malaria. So what you can understand by this study is that we're basically putting a postage stamp in the air and waving it around passively. And whatever is sticking to that, is what we're looking at. And so there's trillions of insects that are on the move. It's not a few insects. So we're finding lots of different insects, plant virus vectors, as well as human vectors of disease. So the latest thing that we're doing is actually taking those mosquitoes down from these high altitudes and now testing them to see if they have any viruses or malaria parasites in them.
Right, because that's if they've had the blood meal, if they are up there, if they're infected with the parasites or any viruses, then when they land, that's the potential to drop all that off wherever they end up.
Well, I think as well, when you start to understand just the numbers that are in that airstream, them coming down and landing is a big...
Yeah, so what is it like, your postage stamps that you're sending up with a big helium balloon? So you've got these sticky traps that are collecting insects while they're up there, but what kind of a sample do you get?
The sample nets are 1 meter by 1 meter, and what you pull down can have grasshoppers. Sometimes bats fly at that height. We sampled up to 240 meters. It's very, very difficult to pull down a helium balloon from 240 meters, and so you need a certain amount, a size of balloon, but you also need a lot of helium. And every time you pull that down, you have to recharge the helium, which can be very difficult to find sometimes. But this is a really interesting study. And I think it changes the way that we view how we have to control mosquito-borne disease, because this is showing that the introductions of many, many diseases could actually be aerial.
Right. And if this is happening in the Sahel, we do know that there are dust storms that bring dust all the way from Africa across the Atlantic to the eastern seaboard of the United States, even within the African continent. How far are any of these insects moving, and what kind of population movement would that be?
That's certainly something that we need to have more data, to actually ascertain, and
fascinating.
Working out where along the clines, we can work out potentially with the wind speeds how far something could go in a certain direction, but that's not necessarily, we're not starting from point zero just because we collect it at this point. That mosquito could have been in its travels for a day or just an hour. So it's very
no idea. You haven't gotten to the point of doing mosquito banding experiments yet, have you?
We need to barcode them more.
Right.
I was just picturing lots of fancy nail polish.
Right, little dots on the backs of the mosquitoes.
How you would find them again.
Yeah, exactly. Yeah,
a little note on them. So if found, please call. Yeah. I wonder if there's some kind of citizen science barcoding experiment that could be embarked on to find more out about this. I mean, it's just so interesting to think about where these insects are distributing. That's fascinating.
Well, I think what we showed, really with the gravidity aspect is that this is a conscious movement. This is a survival strategy for the insects, certainly in the Sahel, and I wonder just how common that is across other regions of the world as well.
As far as disease and mosquitoes, those are the things that we connect in our minds. We like, oh, a mosquito, it's going to not just itch, it's the possibility for any number of viruses or parasites. And depending on where you are, you're going to be familiar with whatever species. But like you mentioned, the Aedes mosquitoes are very well known as disease vectors. What proportion of mosquitoes at large are disease vectors?
So now that we've started to work more with viruses, we're finding that the viruses, unlike malaria, which is only transmitted by Anopheles mosquitoes, viruses are much less strict with where their hosts are. We find a number of taxa able to transmit a lot of different viruses. So when we're talking about Anopheles mosquitoes, around about 70 of the 470 species are known to transmit malaria. But when we started to work on the book, we realized that actually huge numbers have been found positive for viruses. Viruses are somewhat more difficult to ascertain whether the mosquitoes are actually able to transmit the virus that is found in them. So that's another aspect that we have to bear in mind. So certainly in the book, when we were doing the research, we have stuck with associated pathogens because it's very difficult for us to verify that all of those have been tested as competent in the lab. But I think that the number of viruses that we're finding, all these new and novel viruses are very, very common. Whenever we're working in new areas, we tend to find a number of new viruses. We worked recently along the borders of Syria in the refugee camps in the southern borders and also in the northern border with Turkey. And we found a number of new viruses that we described from Turkey just in that time. So we're doing a lot of work on describing new species of mosquitoes and also at the same time looking at the viruses. Yeah,
so you find the mosquitoes and the viruses that are in the mosquitoes, then you say, new virus?
Yes.
What does this mean for humanity? Is that kind of, is this just a mosquito virus or is this something we need to worry about?
That's right. Yeah. And obviously, the mosquitoes can tell us a lot about the environment as well, because now that we're doing molecular analysis, we not only can work out which mosquito species it is with their DNA, but we also have the DNA of the blood meal that they've been taking. So we can work out, which exact host they're feeding on. And also, more interestingly, which of those mosquitoes are feeding on more than one host, because then we have more chance to understand that, which are playing a bigger role in being able to transmit these viruses from wildlife, for example, into humans. Where are those crossover points of these big zoonotic diseases? And obviously, in this COVID pandemic, everyone is very aware of what that means these days. So, we do a lot of biosurveillance work that now includes what we call xenosurveillance. So we look at mosquitoes not only with our entomological head on, but we also look at them as blood bags, which can tell us what is happening, in that environment.
I was hoping you were going to talk about this. I was like, but I want to hear about the blood.
Yes. But the blood, yes.
The blood meal of a mosquito. So, not all mosquitoes actually feed on blood. That's a big misconception. People assume that with mosquitoes, we're always talking about bloodsuckers. And actually, the reason that we have this beautiful image as the first cover of our volume 1 was to show that some mosquitoes, in fact, don't take a blood meal at all. And this is a Toxorhynchites mosquito on a flower. And its proboscis is so bent that it can't actually penetrate skin, even if it was to try. And it feeds on flowers. The reason that mosquitoes feed on blood is to imbibe enough proteins to produce their eggs. So when you have a mosquito that has a carnivorous larval stage, means it can take in all the proteins that it needs to at that stage and can produce eggs from that reserve that it has. So we have the biggest mosquito is the Toxorhynchites, so it's a giant. So tend to think of it as the gentle giant of the mosquito world. And so that was our choice of putting this beautiful image by Larry Reeves on the front cover, was to show that, we're talking about mosquitoes of the world, which means all the diversity of mosquitoes, not just the bloodsuckers or the vectors, which everybody hears about so often.
Right.
So
Well, as humans, we are concerned about the ones that make us itch and give us allergic reactions or cause a disease. I mean, we're just self-centered that way. But
So if Justin were here, he would ask a question. So I'm going to ask it because I'm sure everyone's very curious. He, for a long time, was very much a proponent of specifically those ones that spread diseases. Just wipe them out, right? Just get rid of them.
just get rid of them.
What cause do they serve? What could they possibly do that we'd be losing by getting rid of them? So I'm just kind of curious about the suicide mosquitoes that are being released right now, about that kind of attempt to slow or stop a population of mosquitoes, and just kind of the idea in general of like, do we need all the mosquitoes? I guess is my question.
So, do we need all the mosquitoes? People often ask that. Mosquitoes play a pivotal role in a lot of different facets. So they can play a pivotal role in pollination of flowers. They can play a pivotal role in the food web. If we're talking about bats and birds eat enormous numbers of mosquitoes and other insects in their flights. But, mosquitoes have a larval stage and an immature stage, which is in the water. And so that means that they're food for numerous fish. So if we were to wipe all of the mosquitoes out of the world, we have mosquitoes in almost every habitat that we can possibly imagine. If you have water, you will find a mosquito. You'll find it in the axil of a plant. You'll find it in a tree hole. You'll find it in a spot on the floor, in a puddle that you'll find in Africa. You'll find the most infamous malaria vector in a hoof print that's going to dry up within a few hours, which means its life cycle needs to be that fast that it can go from an egg to a larvae to a pupae and out again before those tiny, tiny puddles of water dry up. So people often say, can't we just get rid of all mosquitoes? For one thing, it's almost impossible. It's almost impossible to find all of those small, tiny little places that you would find mosquitoes. We know that with chikungunya and Zika and dengue outbreaks, the mosquitoes that primarily are involved in that are stegomyia, Aedes stegomyia mosquitoes, and they breed next to us. So if you leave your child's toy out in the backyard and it rains, the mosquitoes will probably be occupying that in a few days' time. If you like flowers and you have flower pots, the mosquitoes will be in any water that you choose to leave around. And that means then that they're also feeding on you and your children, as you're in the garden. So it's actually incredibly hard to get rid of all the mosquitoes in the world. And there's many, many people that are working on vector control and different new techniques and new traps and different baits and repellents. It's a very, very difficult business. If it was easy, it would be done by now.
We would have done it.
There's lots of mosquitoes that we don't need to get rid of. This Toxorhynchites here, I put this as an example page of one of the pages that we have in our book. We have 41 genera of mosquitoes, and each one of them is totally unique and distinct and different. They are geographically bound to the areas, apart from these invasives that we talked about. But they have very particular habitats and ecologies that are linked to that genus. So we have 41 and I believe 172 subgenera. So I'm probably wrong on that number. It changes a lot.
A lot.
But we decided that what we wanted to do in this book was, as well as give a lot of details in the text and the chapters prior to it, we decided that we would do something that was unique, which was to showcase each one of these genera so that people could better understand and identify them more quickly. We looked at the binomics. We looked at whether any of the genera had been associated with any pathogens, what the distribution was, and how we could look at them. So you can see in this picture, anyway, the mosquito has a very curved proboscis, and that's the Toxorhynchites that doesn't feed on man. But we also picked the top 127 vector species around the globe. And so we did this very detailed look and produced these wonderful images through our illustrators, Tina Litwack and Judy Stouffer. They did an amazing job to capture all of the images of all of the characters that we need to identify these mosquitoes. Most people think the mosquitoes are really boring. They're either black and white. Everyone knows the tiger mosquito is black and white, or they are just brown. And actually, the most dangerous ones are just brown, which is why I didn't show them here.
This carnivorous mosquito, you mentioned it's only carnivorous during its larval stage.
Yeah, that's right.
So what do the larvae eat?
They eat other mosquitoes, and sometimes other mosquitoes of the same species themselves. They will usually...
It's a mosquito larva grudge match.
Oh
Yeah.
my.
So these mosquitoes, where we've heard about mosquitoes that are invasive, that move across the world accidentally, man has actually moved some of these mosquitoes into the rice fields. Rice fields are full of mosquitoes. It's a perfect breeding habitat for larval mosquitoes. So in Southeast Asia, and particularly Vietnam, they use a lot of biocontrol using these mosquitoes. Although people see a lot of these mosquitoes, they're very big, they don't bite. So you have, in the larval stage, these mosquitoes are ferocious, and they feed on up to 10 or 15 other larvae in a day. So this can be a very significant reducer of the number of mosquitoes that are there. You can use fish also. There's gambusia fish have been used to control mosquitoes. But obviously, when you drain the rice field, then you kill off the fish. So, mosquitoes eating mosquitoes is somewhat more agreeable to most people.
Yeah. Yeah. What an interesting mosquito biological control. Other mosquitoes.
Yeah.
Just a kind of off-the-wall question. I'm just really dying to know now. How did you feel about bugs as a kid?
I was brought up in the Middle East, in the middle of a desert where there was lots and lots of bugs. I was always very fascinated. Always fascinated.
Lifelong bug interest. That's fantastic. Moving on from our carnivorous mosquitoes, you also have an example here of this Brazilian hairy-lipped mosquito.
This is Trichoposipon. And the reason that I picked this mosquito to look at out of all of many, many others, is that it's super interesting because you find it in the forests, in the neotropics, and it spends some of its time up in the canopy, feeding on the flying squirrels and the monkeys and all of the different animals which are up in the canopy. But you also find it down on the ground biting man. So this is one of those mosquitoes which acts as a zoonotic vector. So it's taking diseases that are in the wildlife and passing it down to us. It has a super neat habit that it does. So basically in the rainforests, obviously, if any of you have been to tropical countries, the downpour is immense. So this mosquito actually breeds in tiny, tiny little pockets of seed pods which are on the ground. And when it lays its eggs in the water, the danger is that it's then going to rain and that its eggs will be washed away. So this particular mosquito will actually guard its eggs. It lays eggs on this tiny seed pod on the ground, and it will stay there and gather the eggs between its legs for up to 24 hours until the eggs actually hatch. Once the eggs are neonates or like small larvae, as soon as a raindrop hits that water, they'll shoot to the bottom. So they'll save themselves. But this is an example of the mother actually looking after the brood, which for me is astounding that a mosquito
parental care
that kind of behavior. Yeah. It's amazing. Her babies are safe until they hatch, and then her job is done. But I think that, doing this book was so eye-opening for me, just seeing all of these incredible evolutionary habits come through, and how you would ever dream that a mosquito would do something like that. There's other mosquitoes that we know just do very, very intricate displays in courtship, which is also fascinating.
Yeah. What did you learn? What was one of the most interesting things you learned as you were going into all these details on all these different species?
Oh, it's so hard. There's another few examples of mosquitoes that we have. And one of the things that I found super interesting was that there is a genus of mosquitoes called Malaya. And they're generally in Southeast Asia. But when you look at them, they don't have a proboscis. They have a club that comes out. And you look at that and you think, well, how on earth does that feed on blood? When you look at every other mosquito that's actually a blood feeder, they have the proboscis, and it's very clear that that's what that is. But here we have a mosquito, and it's tiny. This is, a pinprick of a mosquito, really. But it has this club. And these particular mosquitoes are symbiotic with ants. And the palp that you see turned up at the end of the proboscis is used to drum on the mouthparts of ants. And the ant will then spew forth like a globule of amazingly dense liquid that's, like sugar nectar. And the mosquito will then suck up this offering from the ant, which is incredible.
Amazing.
Excuse me. Wow. Okay, so this is also the second
What does the
mosquito
ant
that
get?
you've shown us that has just the craziest, longest legs.
Yes.
Why do they have these crazy long legs?
The mosquitoes tend to have those legs that they
Legs for
balance with
days.
them. When you see the long legs like that, these are relaxed because they're on pins. So in nature, they would all be crunched up. But it's because we've taken pictures from the National Pins collection.
They still look so long, though.
They are long. They are
Compared to body size, yes.
Yeah.
And I imagine these mosquitoes probably spend most of their time hovering up in air. Would they? Or, yeah.
They would hover. They rest. Often you see mosquitoes resting, and the hind legs will be, waving, sensing the air. That's often how you see them resting on the vegetation. If you look hard enough in a bush, you'll see them.
You mentioned the courtship. Do they use do they do dances and
they do
shake their tail feathers like birds?
It's not all of them do, but there is a genus in Latin America, again in the neotropics, called Sabethes, and the subgenus there has fantastic feathers on their hind legs. They can also have like smaller feathers on their forelegs, but these mosquitoes are incredible. The number of scales that they have, the colors, the diversity of colors is incredible. And we don't really know why they would have all of these different plumages, but they have such heavy legs that when you see them actually flying in the forest, they're so slow you can literally grab them out of the air. So this seems to me not a very good strategy if you're a mosquito to have these heavy plumage. It's been shown that the mosquito, that those feathers are actually involved in the courtship. And so the mosquito has to find a mate, and then they have to go through a set pattern of drumming of the legs and waving of these feathers, and before the copulation can take place. It's been noted that if you chop the feathers off and you introduce a male that has its feathers off, the female will still mate with it. The feathers don't seem to bother it, but the male will not mate with a female that doesn't have the feathers.
Got it. So the female sexual selection, but not male directed. Interesting.
It's really interesting. There's just, so many different small factors that come in. The genus Eretmopodites is another nice mosquito. It's very distinctive. It's almost bright orange in its thorax and then has these silver and black striped abdomens usually. And again, super long legs, as you can see. But these are another one of those mosquitoes that are actually, Eretmopodites is restricted to Africa. It's the only mosquito genus that's wholly restricted to Africa. And it's restricted to the forests in Africa. And it breeds when the monkeys are in the trees and they eat fruit, they split the husk and they throw it to the ground. And that's the breeding habitats of these Eretmopodites and ground snails, just these tiny snails. So you'll find them in there. And again, they're very much linked with being up with the monkeys in the trees. And these are some of the mosquitoes that we're interested in now for transmitting some, forest viruses. And we're interested in looking at these mosquitoes, which are, between wildlife and humans. So in that zoonotic potential to pass viruses, which would otherwise be restricted to non-human primates.
It's fascinating their environmental ecological specialization for breeding. I mean, that you can find little cavities all over the place, but if you're just choosing to live in the cast-off fruit husks and the of a monkey, that's going to limit where you're going to be able to go.
But it also changes the body of the mosquito as well. Where they breed changes the body. We have another genera that's called Udaya. And those mosquitoes, the female will lay an egg singly and capture it with her foot and take her foot and put it into a tiny, tiny insect hole that's been made in bamboo and shoot her leg in there. Or if she can get her abdomen, shoot the egg directly into the tiny hole that's in the bamboo. So if there's a hole in bamboo, there's always water in that compartment. So the mosquito then, breeds really happily. There's no predators there. It's a really safe place for the larvae to grow. But the mosquito has to escape, otherwise it's game over. So you'll actually see that with the genus Udaya, that the thorax is super long and the head is also really, really narrow, and the body shape has totally changed from your regular mosquito so that it allows it to exit from that tiny hole.
Form leads to function.
Yeah. And I just think that when I was doing the research for the book, all of these aspects just came to me so clearly. But there was genus Opifex, and there's only 2 species of these mosquitoes known in the world, and they're restricted to New Zealand. And they look to me like ancient predators. They're so old and scary looking that they should really be in some kind of movie. But on their antenna, they have these huge spikes. Mm-hmm. I was looking and wondering what they could possibly be for. And the mosquitoes are actually, they feed on seabirds. So they have to get between all those feathers and not get damage to their eyes. So I think that this is a protective mechanism for the mosquitoes as they do that. And the arm of the mosquito, or the leg of the mosquito really, but it's its foreleg split into 2. So it's almost like a pincer. It's like the Edward Scissorhands of the mosquito world. I thought, what can this possibly be? This is amazing. Why would they have this structure? And I thought maybe perhaps it's to pinch onto the feathers of these seabirds that they feed on, because obviously the seabirds don't stay still. So the mosquitoes have, they're hitching a ride and they're moving with them. But in fact, it turns out, as I was reading more, that these mosquitoes have been observed the males actually wait on the water for the females to emerge from the pupae to mate with them. And these pincers are for getting the pupal case open far enough that they can put their abdomen in and mate with the female before she's even out of her pupal case.
Oh my!
Wow!
The sexual predator of the mosquito world.
Hello.
And all of these crazy things that we found on the way through of doing the diversity of mosquitoes. Amazing. Just makes you want to learn everything about every species that's there, So it was a lot of fun working through all these amazing things. And, as we're finishing up with the book, it was a great paper that was written by colleagues here in US on Uranitania serifina. And they determined that this particular mosquito here in USA feeds on earthworms. Now,
earthworms.
I have several small children, and one of our games is go find a worm. Sometimes we're lucky, sometimes we're not. But for a mosquito to be able to find earthworms as their primary target, when we think about mosquitoes, we think about heat, CO2, all of the different attractants that are pulling those mosquitoes to their host. But what could possibly be attracting a mosquito to a cold-blooded worm?
And earthworms also are underground.
Yeah, that's what I was going to say.
I mean, until it rains, and maybe when you have a big rain, they end up at the surface. Yeah.
Yeah.
Fascinating.
So I think that we have so much more to learn. But what we tried to do with this book, certainly Dan and Rick are both retired, and between us, we have 100 years of experience with mosquitoes. So it was a huge, huge honor for me to work together with Rick and Dan on this book and really to lay down for the future the knowledge that they have and that they have accumulated over their working lives. The second volume of the book, and I do laugh because it's 12.5 pounds worth of book. Which is ridiculous.
It is a massive, the work that has gone, I am so astounded and impressed by the amount of work. This is 100 years worth of knowledge put into to 2 massive volumes. You must be so proud of the achievement. It's just amazing.
It's amazing to finally see it. We've worked long hours, and, weekends and nights and middle of the night for almost 7 years pulling all this data together. So, and one of the things with mosquitoes is that you're never quite finished. You know, as you have to draw a line in the sand and say, this is where we have to stop, because otherwise this book is never being published. We need to publish this and share the knowledge and ask our colleagues to help contribute to maintaining that knowledge. So at WRBU, we have our website. It's wrbu.si.edu. And on the website, we'll be keeping a list of the updates to anything that changes in the book. The catalogue is the volume, the second volume of the book. And this is really the labor of love of Rick for forever. He started an online catalogue and has maintained that since the 1990s. And this is the succinct, important taxonomic works that has been done for every single species in the world. So every entry that's there, every name that it's been previously given, its distribution, where you can find the original type specimen, who described it, what its name means. It's very nicely done. Johns Hopkins Press have done an amazing job of laying out the catalogue, and they've helped us by giving us colours, stripes of colours for different genera, which is always really helpful. But basically, the last time that a taxonomic catalogue was attempted for mosquitoes was in 1977, so almost 50 years. So it was long overdue for an update, but it was a marathon effort to try
and I'm sure so much has been learned and so many more species named, discovered since the 1970s when that last catalog came out. So you're probably adding exponentially to the volume of work that's out there.
Well, it's certainly, just for the references for that work is more than 200 printed pages. So just the reference list, which is amazing. But yeah,
I respect you for being able to maintain a reference list that's that long and complex.
I'm sure that there's definitely a good number of references that we didn't pick up at the time. You know, that we did absolutely the very best that we could, but I think that this is a work that's more than three man's work. So moving forward, we really, really encourage the rest of the community to help us keep this up to date and to point out any things that we've missed or any errors that, you know, creep in. It's a work in progress, and as we move forward with identifying all these cryptic taxa with molecular means, you know, then you end up splitting species that were before, and we're going to have to, as a community, work out how we deal with all of those moving forward as well. We just simply do not have enough taxonomists to name every single species that we find. There's a global taxonomic impediment in every field, and mosquitoes is no different. So, but not everybody.
That's why everybody's naming everything after David Attenborough now.
David Attenborough was actually instrumental in starting my path on the mosquito barcoding initiative. Oh, wow. I was at the Natural History Museum in London, and he helped with a donor's dinner, and that set the path for the mosquito barcoding initiative. So
wow,
we were very privileged to have met him and have him influenced.
Yeah, that's fantastic. He's done such amazing work. I'm not saying that he shouldn't have species named after him. I just feel like
there's a lot.
There's a lot.
Are you looking for one after you? Are you? We have a lot. Right. We found a lot.
Because I need to be a blood-sucking mosquito.
Well, our latest manuscript is actually a mosquito named after my mum. So
that's fantastic.
A fun one. Yeah.
Mom, I love you. I named a mosquito after you.
I'm not sure how she'll take it, but we'll see.
Well, from you, that's a very big compliment.
Yeah, probably. So wonderful. What is one thing about mosquitoes that you hope people will know and understand moving forward?
I think for me, I would like people to appreciate the diversity within mosquitoes, not just wholly to regard them as disease vectors, but to hopefully better understand the role that they play within the ecosystem, and to appreciate them for the uniqueness that evolutionary time has generated within mosquitoes. I think that they're a fascinating group, and I think we can learn a lot from them. Personally, I love the fact that they transmit all these fabulous viruses. And I'm super interested by all of that. But I do hope that, people will have an appreciation for mosquitoes that maybe they didn't before they look at our book. I think that some of the data chapters at the start are really detailed and very nice insights into biogeography and copulation of mosquitoes, development, oviposition, what stimulates the females to oviposit in the different habitats that they have.
Thank you so much for joining us tonight. So your book, Mosquitoes of the World, Volumes 1 and 2, it is available now. You hope that entomologists, that insect lovers, maybe teachers, maybe anyone interested in mosquitoes will take a look. Where can people find you? At the WRBU?
Yes, at WRBU. So we have on the website, you'll see that there's a contact, and you can email us through that contact, and we'll pick that up and respond.
Before tonight, I knew a bit about mosquitoes, and now my eyes are open to just the amazing diversity of biology of what mosquitoes have to offer. A lot of blood bags and blood sucking, but there's more. And I do appreciate that. So thank you. Thank you so much for joining us.
Dangly legs, lots of long, interesting proboscises. Is that the plural?
Proboscises. Yeah, I guess they only ever have one. So it's just a proboscis.
It sounds weird, but it seems like it would be a very cool coffee table book in a funny way.
We hope that people will use it in lots of different ways. Definitely those people who are working with mosquitoes, that volume 2 will become their new Bible. That's what we're hoping. It's certainly the way that I've used the last catalogue that was 50 years old. I've looked at it every day. So I'm glad to have an updated version.
Thank you so much once again for joining us tonight. Maybe we can get you back on to talk about mosquitoes at some point again in the future. It was just fantastic. Thank you so much.
Thank you.
And everybody, we are going to take a very quick break. Come back in just a second with more This Week in Science. This is This Week in Science. Thank you for listening. If you're enjoying the show, please help TWIS grow by suggesting a friend to listen to it today. Yeah, get a friend to subscribe today. That'd be great. Back to the show. It is time for this part of the show that we love. That it's not about poop tonight, I don't think.
No, the poop was before.
Right. Okay.
This is all about carbon dioxide.
Oh, that's right. This is the carbon dioxide climate change time. It's time for Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. Wanna hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
With Blair.
Thank you. So I want to talk about coral bleaching. I don't bring it up in the Animal Corner very often because there's not usually new news. It's just kind of like, hey, corals are messed up. Hey, did you know that warming oceans are bad for corals? It's just that kind of over and over again. Oh, yeah. Another coral reef bleached again. Oh, darn. So anyway, this week we actually have some very interesting coral bleaching news that it might impact future management of corals and how we can predict the response of corals to ocean warming. So this is largely an international effort, but this is mainly out of École Polytechnique Fédérale de Lausanne.
Very nicely done.
Le français. Oui. Using that French minor every once in a while just to pronounce a word.
That's right.
I'm so proud. Anyway, so as we know, corals bleach when the water gets too hot and they expel their photosynthetic algae, their symbiotic algae that lives inside of them. Because the reason coral reefs are so beautiful and so pristine looking is that actually it's kind of garbage habitat because the water is so clear. That means there's no nutrients in it. So the corals, which are actually little animals that almost look like teeny tiny anemones, that then secrete this calcium carbonate skeleton that turns into these kind of giant apartment buildings, is kind of how I always think of it, where all these little anemone creatures live in. So coral is an animal. I always have to bring that up because people don't always make that connection. But so they are this colonial animal that then they're filter feeders, but as I said, the water's so clean they can't really eat too much just via their little teeny tiny anemone shaped bodies. So instead, they have these photosynthetic algae, zooxanthellae, depending on what you want to call it. And they are responsible for the amazing colors that corals are. And they are using their photosynthetic processes to then deliver nutrients to the coral. And they are provided a home via the coral. So all that together, they have this symbiotic relationship. But when the water gets too hot too fast, the coral freak out, and they kick all the algae out. That's called coral bleaching, because they all look kind of like white and pale because all of these beautifully colored algae have exited. Now, over the past 35 years, there have been a bunch of different heat waves that we've measured. And during these stressed moments of warmer temperatures, they kick these guys out. And, it's not necessarily a death sentence. They can bring the algae back into their bodies if they calm down a little bit. Mm-hmm. They get a little bit more used to the situation. Maybe the hot spell doesn't last as long. They're able to bring it back in, and then they can survive. So a coral bleaching is not a death sentence to a reef. However, the expectation was that the stressed coral just kind of, they don't know how to function anymore. And then they just kick them all out. They're just like, I'm stressed out, get out. Or that actually the algae is somehow not providing them the nutrients. And so they kick them out for that reason. They're basically a victim. Like, you're not
paying your way.
Get out of here.
Get out.
But this new study looked at kind of specifically the order of events here and what causes corals to bleach. And they found some brand new information. So the algae, which is stressed by warmer temperatures, they release compounds that are toxic to the coral, which then prompts the coral to expel them. Which that much we knew. So there's some sort of toxic exchange that then the coral goes, well, fine, then get out. But this new study found how actually the coral starts to suffer from hunger before the algae are expelled. So the algae stop providing nutrients. They're still inside the tissue. Then the coral's already stressed and hungry. Then the algae releases toxic compounds. And then they say, that's it, get out. So it's actually a compounding of multiple factors here that cause them to do this final extreme gesture of kicking out the algae. So the roots of the problem are deeper than was previously thought. It involves breakdown of metabolic exchanges in between the two organisms. And so this is all discovered over a year studying coral reefs in the wild and then replicating these environmental conditions in aquariums so that they could investigate very specifically the data that was happening in these corals when they're bleached. So one of the lead researchers says, based on what we found, we can determine which environmental conditions, other than temperature, such as water quality, stress the corals in a reef, and use this information to predict whether the reef will bleach. So first of all, you can figure out where to expect bleaching, which is helpful. Mm-hmm. Then our findings can also be used to identify corals that are particularly resistant to bleaching. So that can help scientists determine which reef should be most protected because they have the best chance of survival in a warming world.
And also what's going on in a particular reef that's protecting them. Why is this reef so much more resilient than this other reef?
Right. Well, and we've done stories on the show before about transplanting more kind of hardy corals into bleached areas. So that's something that you could do here as well with this information. But it's interesting to
Give
see
them probiotics.
Yes, exactly. Exactly. So it's interesting to see some of the actual mechanisms at play here. Because, I always just kind of pictured the crow going, I'm hot, get out!
Yeah, it's like, no more, the blankets are too hot, I can't do it, get out of my bed. There we go.
I can't focus on you and this, you gotta go.
But it's just, there's a lot of stress.
Mm-hmm.
The corals, they have to deal with a lot of stress. And there's a lot, they're not just one kind of stress, many kinds of stress. We just have to know what those are and what different thresholds may be affecting where they are at a particular time. Yeah.
Yep, absolutely. So that's from warming waters, which of course come from a buildup in carbon dioxide in the atmosphere.
Mm-hmm.
But there's also the piece of the carbon dioxide that then gets absorbed by the oceans, which is somewhere between a third and half of the carbon dioxide that we emit ends up in the oceans.
Mm-hmm.
Closer to a third, but there's a lot of different numbers for it. But ultimately, that has a lot of impacts on aquatic animals. And with fish in particular, last year we reported on the fact that some fish are shrinking in higher CO2 levels.
Yep.
Some fish, their senses are impacted by CO2 levels. There's lots of different ways animals can be impacted by CO2 in the ocean. But this new study from University of Adelaide found that some species of fish actually have higher reproductive capacity due to larger sex organs under more acidic oceans.
Wait, what?
Yes.
So the oceans are acidifying and they're having bigger gonads?
Correct.
Even though they're shrinking.
Well, these guys aren't shrinking. So
okay.
some fish are shrinking, but these guys are not seeing any deleterious effects that they have found. Asterisk, right? That they have found.
Asterisk. Yes.
Disclaimer. That they have found. So at this time, they haven't found any negative impacts on these fish. These are triple fins. They're a species of temperate fish, and both males and females had larger gonads under conditions of ocean acidification, increased carbon dioxide level in the ocean. This meant increased egg and sperm production, and therefore more offspring. They also looked after them better, enhancing the chances of reproductive success. They used natural volcanic CO2 underwater seeps to compare ecosystems with the levels of carbon dioxide predicted for the end of this century, with fish living under today's quote-unquote normal levels of CO2. And I love that now that we have to put normal in quotes when we're talking about today's CO2 levels because they're bonkers already.
Not normal.
But so that's an interesting variable here that I will just point out is this wasn't a highly controlled laboratory experiment. This was naturally occurring fish that they recorded in 2 different environments to mimic different carbon dioxide levels that could be the standard in the ocean. So there could be other variables going on. I'm just throwing that out there. So I don't, but anyway, so they saw no negative effects of ocean acidification. They did not appear to have the larger gonads did not appear to have a physiological cost of any sort. There was no trade-off that they could see. But the way that happened is it looked like males were eating more. They showed intensified foraging, and their prey was more abundant because of increased biomass of algae that grows under elevated carbon dioxide. So that could maybe track. More algae means more feeding, which means bigger gonads, I guess. But the females, they didn't eat more at all. They just moved less. They had reduced activity levels and preserved their energy that they invested into larger ovaries. So they had more babies in general. And there were more observations of parents nurturing egg nests, which could also increase offspring. So I bring this up only because there's a lot of conversation when you talk about the future and climate change, and the phrase often comes up, winners and losers, right? And so this is a situation where you could see this as these fish, the triple fin, could be a winner in climate change. However, the thing that I will then just throw out there for you to consider, humble listener, is that, as I always try to say, it's all about system, about ecosystem, about entire habitats and food webs. And we don't know if an entire clade, an entire family tree, an entire type of fish, or even just one species here, one species there, suddenly performed better under climate change, how that would impact the ocean environment as a whole. Right. Is that going to impact the food web? Is that going to impact the kind of distribution of biomass? Is that going to impact water quality? Right? So if they're eating a bunch more algae, is that going to spike CO2 even more because you're eating the stuff that usually processes CO2, right? So there's, just because on its face you see what you might want to call good news and that an animal is winning in the climate change game, they could actually boom and bust as a result of higher CO2 levels. Just like when you take out the wolves from, where was it, from Yellowstone, right?
Right.
The deer population explodes, and then they all get sick and start to die.
Natural population control.
There's population control, and there's all these other factors. So I think on its face, this is very interesting. It's something to watch for sure, because this is a good reminder that every animal, every species is going to react differently to a chemical change in the oceans. That's a pretty drastic thing to just change real quickly all at once. But there's a domino effect here that we can't see the end of yet.
Yeah, but it does seem as though, so these fish are, their gonads are growing, they're having more young, laying more eggs, they're being more protective over their eggs. So yeah, they're having more offspring, but perhaps because of the ocean acidification, there is less food. And so they're having more young because there's a lower chance of survival. Or, there are lots of different ways that it could trade off.
Right. And, I'm sure that these researchers did a really good job of accounting for other things. But I will just throw out there again, this was a naturally occurring seepage that had CO2 levels. So was there temperature difference? Was there mineral differences? Was the terrain different? I'm just throwing it out there that this is an interesting initial look, but a controlled study in a lab would, I think, give us more clear results of the effect of CO2 on the species. Right.
And was there time for adaptation to take place in this particular population in the wild versus, a manipulated laboratory experiment where you can switch carbon dioxide levels very rapidly?
Totally. And that's a fair point, right? Are they genetically dissimilar, these two populations, or are they commingling and it's just the ones that live over there have bigger gonads? If they're genetically different, then that might mean that they have been changing in this way for a long time. And then will ocean acidification happen too quickly for this to matter at all?
Right. Right. I mean, that's the big argument, is that this is all changing so quickly that only the very rapidly adapting species are going to be able to keep up, catch up.
Yep.
Yeah. Yeah. Oh, okay. Let me talk about brains.
Please.
Can I take you away from some animals and talk about brains?
I would love it.
Awesome. Let's talk about, well, oh no, I know what I want to talk about first. This brain, this is a story about women. This whole story right now. I want to talk about women. Blair, have you heard the idea that women and our menstrual cycles, our reproductive cycles, synchronize to the moon and the lunar cycle?
I have heard this. Yes.
Many times. And every once in a while you're like, oh, look, the moon, and here I am, and maybe. And then you're like, the next month you're like, that didn't work at all. So there are these questions we don't know, and people have been studying this for years, decades, trying to determine whether or not women's menstrual cycles are tied to the lunar cycle. Mm-hmm. We've had studies that have been short studies with lots of women where they've only looked at like 1 or 2 cycles. We've had our cycles where they've followed individual women. They've studied big groups, all sorts of things. Most of the research, though, has not really found a link. And there's a new study that's out in Science Advances this month that is looking into this question of whether or not women's menstrual cycles are synchronized to the moon. And you know what? The question is still there. They didn't find an answer either. So these yeah.
I haven't heard the actual story really yet, but I'm already having problems with this because other mammals aren't on lunar cycles.
But there are aquatic organisms that are, so why not women?
But why, out of all the mammals, why would humans be the one? They're like, oh yeah, I gotta link my period to the moon.
So apparently, one of the data sets that they looked at to support this study is wild gorillas, that wild gorillas appear to be synchronized to the moon and the cycles of the moon. Blair, you gave me a funny look.
Okay, all right.
I didn't write this paper. I'm just reporting on it.
I've never heard such a thing, and I'm thirsty for knowledge. So please continue.
Let me continue. Yeah, so they looked at 22 women. So it's a very small sample set. But of these women, they kept long-term records. And so they had records up to 32 years in some cases of menstrual cycles. And then they tried to look over time at how these cycles lined up with moon cycles. And, you know, 22 people really is not a very large sample set, which is, I'm going to say, the big limitation of this study. Any results that come from this are mostly anecdotal. This is like a pilot study. They really need to have more long-term sample sets.
I feel like it would be very easy to get this data. I feel like lots of people, especially now, use period trackers on their phones.
Yes. And that's part of the study. That's what they say at the end of their paper, is that what we need is new long-term data sets, and maybe technology can help. So this is a direction that they are trying to look. But when they did line up women's cycles, they discovered that older women seem to have less synchronization with the moon than younger women. So women over 35, less synchronized than younger women. They think that it's because older women have been affected by our modern technology and light and devices and other things longer than younger women. And so maybe that's what's driven older women's cycles out of sync with the lunar cycles. But they did find that several individuals within their sample did not just line up based on the light cycle of full moon to new moon and, you know, the phases of the moon, but seemed to line up also with a gravitational aspect of the moon, so that when the moon over several years is in its closest position, so its gravitational effects of the earth on the earth are the strongest, several of the women in multiple years did line up with their cycles with the moon in those instances. So they suggest that this correlation, it's not causation, that there's this correlation between possibly gravitational effects of the moon, also the light of the moon during the full moon. Could maybe, but they still don't know. And they need to do more study. But they do want to do a directed study that takes into account our modern society and the devices that we use, the blue light that we're affected by constantly from our screens and our small screens, and how late night light affects us. Unless
So
it's
I
light from
get
the moon.
all of that because
Mhm.
we know blue light impacts circadian rhythms.
Yes.
So why not?
And circadian rhythms affect hormonal rhythms and all this kind of stuff.
Yeah.
So there's
Sure.
this whole thing has always had some potential in it somewhere, but nobody's ever really found that link. And so the big take-home message from this study is that they hypothesize that in pre-modern times, women were likely to have been synchronized to the moon more often than not. But because of modern society and light and staying up late and changing our circadian rhythms through just living today, that doing that has taken us away from that synchronization.
Or, if I may,
You may go for it.
a menstrual cycle is supposed to be approximately 28 days. A lunar cycle is, I just looked it up,
27.35.
29.5 or something like that. Yeah, so it's right around it.
Mm-hmm.
And also, just generally speaking, 35 is when you're supposed to be like less reproductively viable.
You start
as a
becoming
woman,
you
right?
start becoming less reproductively viable. Your menstrual cycle starts becoming less and less predictable. And
yeah. So it could just be a funny coincidence that they have about the same period of length, no pun intended, and then things get dodgy after 35, which is all it's like that.
Yep. I have many issues with this study. I love the fact that they've done these long-term records with women, because how fascinating to be able to track somebody for 30 years for the majority of their reproductive ability. And that in itself is very cool.
Yeah, I can tell you, I'm sitting, not to get too personal, but I'm sitting on, I think, 8 years of data on my phone right now that I'd be happy to provide to a scientist. So,
right.
I'm not the only one. So, like we talked about before, I feel like this data set could get beefed pretty quickly. Yeah, absolutely.
And then there's also women who are taking contraceptives or others. How do those affect the cycle? By managing it. So there's so many things, but this question, it's ongoing. Now let's talk about brains, because brains are something that I just really love. Decisions are very hard to make, and it would be really cool if we could track decision making in the brain, and maybe even be able to know the decisions that people are going to make ahead of them making it. Especially for, not autonomous, but for human brain-controlled interfaces. So say you're controlling a drone with your brain. You're like in the military and you're an Air Force pilot, but your plane is over the Middle East somewhere, right? So if that's the case and you're not using your hands, because it takes a little bit longer for the instructions, the decision in your brain of the action you're going to take to go from, I'm going to make this decision, to actually going to your hand to move a joystick or hit a button. If we had a way to know the decisions that people are going to make ahead of time, those decisions and actions could occur more quickly. This could happen for all sorts of brain-controlled devices in the future. Other concerns, though, are interfacing with your phone, with your computer. And what if it knows what you want before you know you want it? No, I didn't mean to buy that Amazon thingy.
No thanks. Unsubscribe.
Unsubscribe. Hopefully my brain would be unsubscribing me from all sorts of stuff, because that's the first thing. And then I second guess myself. But the process of deliberation, the second guessing, this goes back and forth. And so as the brain is taking in information, it's going to be weighing the information that's coming in before making that decision, coming to the final decision. And so researchers put together a a new real-time brain monitoring system, which they have tested on monkeys. Whoa. Something just turned on here, and I suddenly have a video playing somewhere, and it's very loud in my brain. There you are. Go away, advertisement. Oh my God, what's going on? That was crazy. You can't hear it, and I sound like a crazy person. That was just weird. Okay, so these researchers, they were tracking in the dorsomedial prefrontal cortex of monkeys to be able to track these monkeys as they made a decision. And the decision they had to make was which direction a set of little dots was going to go. And the information that was coming in ahead of time were these little tiny dots were giving them information as to which they should choose, right or left. So a bunch of dots coming in, and if most of the dots are over on the right, then the monkey was supposed to choose right. If most of the dots are over on the left, the monkey's supposed to choose left, something like that. So the monkey's brain is tracking the position and the timing of these dots appearing on a screen. And as that information was coming in, the researchers were able to view the brain's activity on a screen. And the researcher said, this researcher who is involved said, I was just looking at the decoded activity trace on the screen, not knowing which way the dots were moving or what the monkey was doing. And I could tell the lab manager he's going to choose right, seconds before the monkey initiated the movement to report the same choice. Seconds ahead of time. And the researcher, his name's Diogo Peixoto, he is a co-author on the paper and said, I would get it right 80 to 90% of the time. And that cemented in my mind that it was working. So it's this real-time tracking of the brain's decision-making process could potentially open up a whole new window, not just in decision-making, but in a lot of cognitive aspects. Neuroscience studies looking at all sorts of activity in the brain could use this new technique to be able to predict what the next action, cognitive choice within the brain could potentially be. Now,
it makes me think about impulse control a little bit.
Yeah.
Because I mean, I know especially like if I really want that dessert, I'll think that I'm deciding for a while, but I've made up my mind.
Oh, I know what I want.
I know what I'm gonna get, and it's not gonna be the piece of fruit. It's gonna be the chocolate.
Chocolate. It's gonna be the chocolate. Yes. So the decision process, though, that's going on in the brain, where the researcher is able to look at this output, the decoded output of the activity, is the decoded output. The researcher is able to go, I can see what the monkey's going to do before the monkey's going to do it. But in the brain, though, there's multiple neurons that are responding to different types of information, different aspects of the environment, and all that information needs to be collated. Those electrical signals the noise needs to be taken out, and the signal actually needs to be processed to be able to lead to the output that they can actually use. And one of the things that they wanted to mess with were those dots that the video screen was showing to the monkey. And in one of the experiments, they decided to see how many dots they could add to the experiment before the monkey got distracted. And then while they were adding those dots, they added dots in this so that they weren't bright dots on the screen, but just below noticeable threshold where the eye could make out the light, but it wasn't something that the brain would be, I see a dot on the screen, where it's below the conscious threshold. It's like a subliminal dot on the screen. And the researchers tried to use these subliminal dots to bias the monkey's choices. And they were able to bias the monkey's choices. But if they added those dots in after the monkey's brain activity, after the monkey had already taken in enough information to get on the direction of making a choice, that choice was already dialed in, and that subliminal subliminal stimulus didn't have any extra effect. So there is a level at which certainty happens. There's a threshold for that. But the part of this that bothers me is they were using subliminal stimuli to influence the monkey's decision, and they were able to do it, and they were able to see that they were able to do it before the monkey made the choice that was subliminally influenced. So what does this mean? Yeah. Like, we know what choice we want you to make. And so because we can read your brain activity, we're going to give you a particular input to lead you down that road to making that choice. So could be used in a very interesting way, advertising.
Push me towards that chocolate even harder.
Yes. No, that choice was already made, Blair.
Yeah, that's true. It's important, right, to know in the brain when you make the decision, because there's definitely human applications I could think of there in a lot of ways, but especially like intervention in bad behavior or detrimental behavior is really what I mean to say. So if you're trying to figure out what's going on in somebody's brain that's causing them to do something that's hurting themselves, might be helpful to know when that decision is being made and where and how. So
yeah, that is a great way to look at it. Anyway, those are my choices for stories tonight. Those are all the things that I wanted to talk about. For those of you out there who have questions about COVID or anything else that, ask the TWIS team a question. You want to know what our favorite movies are? Ask us that. You want to ask us a question about science? Ask us a science question. We're here for you. If you have questions, send us an email. My email, Kirsten@thisweekinscience.com, or a message over on Facebook. You can go to the This Week in Science Facebook page, leave a message there. Also, our TWIS Twitter account, you can leave us a message there. If you want to ask a question over there, we can copy and paste it in here.
There was actually one question that came up a bunch during our interview that I think we can answer in like one sentence, which is, can mosquitoes give you COVID? And we've talked about this on the show before. The understanding is no, they cannot, because it is not a bloodborne transmission. It is a respiratory droplet, an aerosolized droplet transmission. So it's from the mouth and the nose.
And the nose, as opposed to the blood.
Yes.
Yeah. So the method of transmission makes a difference.
Yeah.
Yeah. And then I guess final comments. I wanted to comment briefly on people's excitement around ivermectin, which is a potential therapy for and maybe prophylactic for COVID-19. And there are a lot of YouTube videos about it right now, and Facebook pages and Twitter tweets, and people are very excited about it. And the NSF and the World Health Organization, I believe, have opened it up to use because they've determined that they don't think it'll do harm, but they still don't know if it'll do any good. And yes, there are thousands of studies available on ivermectin and how it works in cells and also how it works in the human body, because it is an antifungal that has been used for years, but it's not an antiviral. It is an antifungal. So we know how it works with respect to certain fungus infections, fungal infections. The thing is, we know it works in human cells in a petri dish. We know it's worked like in some animals, but we haven't done the studies yet that are clinically randomized controlled studies in large samples of people to know how it affects COVID-19, SARS-CoV-2. So just like the hydroxychloroquine, just like and yes, any number of wonder drugs that people want to jump on the bandwagon for. The research is out there. The researchers are doing the studies. So have patience and be a bit skeptical of amazing claims. But, it could be something that could help people, but we don't know enough yet. So
I think this is why the real science gets muddled in media, right? Is that, you have this amazing claim that this is going to save your life, or you have this amazing claim that this other thing is going to kill you. And then in reality, we have we have these vaccines. They're 95% effective. You might feel crappy for a day after you get your second dose, but then you should be pretty well protected. But the thing is, you might still be able to spread it to someone else. So still wear a mask. So it's the fact that it's
process.
The real,
it's not as easy.
Thing is squishy and imperfect in its own way. That's how you can tell it's real, right? Is that you're getting the whole story. It would be nice to be able to say, this will kill you, this will save your life. But it's much more complicated than that.
And it usually is. And if it seems too easy, might be a reason. So yeah. All right, everybody. I think we have come to the end of our show. Loved our guest tonight. Big thanks for Dr. Yvonne Linton for joining us tonight. Her book, Mosquitoes of the World, with her co-authors, if you are interested, is available now. Thank you all for listening. I would love to shout out to a few people. Shout outs to Fada. Thank you so much for helping with social media and show notes and the YouTube chat room. Gord, thank you for manning our main chat room. Identity 4, thank you for recording the show. And as always, thank you to our Patreon sponsors and to the Burroughs Welcome Fund. Thank you to Woody MS, Andre Bassett, Chris Wozniak, Dave Bunn, Vegard Shefstad, Hal Schneider, Donathan Stiles, a.k.a. Don Stylo, John Scioli, Guillaume, John Lee, Ali Coffin, Gaurav Sharma, Shubru, Sarah Forfar, Darwin Hannon, Donald Mundus, Stephen Albaran, Daryl Myshak, Stu Pollock, Andrew Swanson, Fred S104, Sky Luke, Paul Ronovich, Bentley the Translator, Big Nell, Kevin Reardon, Noodles, Jack, Brian Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hessenflow, Gene Tellier, Steve Leisman, a.k.a. Zima, Ken Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard, Brendan Minish, Melizond, Johnny Grindley, Kevin Railsback, Flying Out, Richard Porter, Christopher Dryo, Mark Massaros, Artyom, Greg Briggs, John Atwood, 2020 can bite my shiny metal. Arrs. Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Matt Sutter, Philip Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavez, Alex Wilson, John Ratnaswamy, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Pecoraro, Gary S., Tony Steele, Ulysses Adkins, Brian Condren, Jason Roberts, and Dave Friedel. Thank you all for your support on Patreon. We can't do this without you. And if you would like to support us on Patreon, make sure you head over to TWIS.org and click on the Patreon link. Support, and you can choose your level of support over there. I want to read your name. Make our list longer. On next week's show, it's gonna be the beginning of February already. Oh my goodness. I don't have a guest yet, but I'm working on things. And we will be back on Wednesday at 8 p.m. broadcasting live from our YouTube, Facebook, and Twitch channels and at twist.org/live.
Hey, do you want to listen to us as a podcast? Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends maybe to subscribe as well, perhaps.
Perhaps subscribe. For more information on anything you've heard here today, show notes and links to stories will be available on our website, twist.org. And you, yes, you can sign up for our newsletter.
You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twist.org. Just be sure to put TWIS, TWIS, in the subject line, or your email will become a blood meal of a mosquito.
No. You can also ping us on Twitter, where we're also at Twisinus. Twisinus? No. Twist Science. Dr. Kiki at Jacksonfly at Blair's Menagerie. We love your feedback. And if there's a topic you want us to cover or address, a suggestion for an interview, a haiku, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news. And if you've learned anything at all from tonight's show, remember,
it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. Jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. So how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say. And if you've learned anything from the words that we've said, then please just remember it's all in your head. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.